Hello, good afternoon, everybody. This is Krishna Kalluri, alternate counselor of the Southern California section of the American Chemical Society, SCALAX. Our SCALAX, in collaboration with the Georgia local section of the ACS, submitted a proposal for an innovative projects grant. They call it the IPG grant, to the local section activities committee, LSAC, and we are awarded that. And the proposal contained the organization of symposia relating to biosensors, their design, construction, and the use of the chemical technology, like the analytical, I should say, technology like optical, electrochemical, piezoelectric, and other forms of detection of the interaction between an analyte and a biomolecule. And we got that grant, and under the auspices of that grant, this is the first seminar we are organizing, which would fall under the optical biosensors classification. I would like to introduce tonight's speaker, Zhang. Dr. Zhang received her bachelor's degree from Tsinghua University and her Ph.D. in chemistry from University of Chicago in 2000. After completing her postdoctoral work in the laboratory of the late Roger Sheehan at UC San Diego, she joined the faculty of Johns Hopkins University School of Medicine in 2003. She was promoted to professor of pharmacology in 2013, continued there until 2015 when she moved back to UC San Diego, and she is currently professor and vice chair in the Department of Pharmacology. Research in her lab focuses on developing enabling technologies to probe the active molecules in their native environment and characterizing how these active molecules change in diseases, including cancer. Dr. Zhang is a recipient of several awards. In 2009, the Biophysical Society Margaret Oakley Day of Award, the NIH Director's Pioneer Award. Then in 2012, the John J. Abel Award in Pharmacology. In 2012, Pfizer Award in Enzyme Chemistry, the NCI Outstanding Investigator Award in 2015 and 22, Robert Raffaello Career Achievement Award in Pharmacology 2022, Protein Society Christian B. Ansmithson Award in 2022, and Biophysical Society Caroline Cohen Innovation Award from 2023. She was elected as a fellow of the American Association for the Advancement of Science, AAAS, in 2014, a fellow of the American Institute of Medical and Biological Engineering in 2019, and a fellow of American Society for Pharmacology and Experimental Therapeutics in 2021. Dr. Zhang also received UC San Diego Chancellor's Award for Excellence in Postdoctoral Scholar Mentoring in 2019 and UC San Diego Jacobs School of Engineering Outstanding Graduate Student Mentoring Award in 2022. She currently serves on the Scientific Advisory Board for Max Planck Institute for Medical Research, on the External Advisory Board for Biophysics and of the Johns Hopkins University, and on the Basic Sciences External Advisory Council of Vanderbilt University. I'm sorry I took away a lot of time introducing Dr. Zhang because her biodata is so extensive, I can't help taking that time, less than that. So I will pass it on to Dr. Jin Zhang to present her seminar now. Over to Dr. Zhang.
Thank you, Krishna. Thank you for the invitation and also for the kind introduction. I guess I'm honored to give the first seminar for this symposium, and love to interact more with the Southern California local section of the ACS. So today I'm going to talk about some of our recent work on building optical biosensors and how to use them to look at self-signaling. So for my lab, I guess for the last 22 years, my lab has been interested in understanding how the cell organizes biochemical activities to achieve very specific functions. At the molecular level, these biochemical activities, such as shown here, represented by kinase and phosphatase activities, are underlying every aspect of cellular function. And these biochemical activities are also connected, integrated in a complex network like this. Each of these connections is made through protein-protein interactions, small molecule protein interactions, RNA protein interactions, and also enzymatic reactions, these biochemical activities. And these complex networks are also embedded in the spatial temporal framework of a living cell, being constantly modulated. That's why we've been interested in building these optical biosensors to go into the native context of these biomolecules to probe their properties, behaviors, regulation, and function. And from our work and work from many other labs in the field, we recently came up with this hypothesis. We hypothesize cellular biochemical activities are not floating inside the cells. They're actually very well organized. They're spatially organized into an activity architecture. This is our best attempt to, I will change to, can I change to the laser pointer? This is our best attempt to really depict this activity architecture. And we further hypothesize disease states such as cancer reflect the reorganization and restructuring of this activity architecture. So we've been interested in probing this activity architecture across the different scales, from molecular to nano micro scale to subcellular and all the way to the whole organism level. I will start the talk by introducing some of the biosensors that we have been building in the lab. Shown here is the general design of our FRET-based kinase activity reporters. This is the slide that I have been using for 22 years because we started the lab with this design to try to build these kinase activity reporters, which we call CARs. These CARs allow us to track dynamic kinase activities in different living systems. As you can see, we're basically stitching quite a few components together and use that as a fluorescent biosensor. And two of these components you probably recognize are a pair of fluorescent proteins, shown here represented by cyan and yellow fluorescent proteins. And these two fluorescent proteins have the appropriate spectral overlap. So when they get close together, this yellow guy steals the energy from the excited cyan and glows at its own wavelength. And of course, the efficiency of this energy transfer process, FRET, is dependent on the distance and the relative orientation between the two fluorophores. So together, they serve as reporting unit of our biosensor. And we spend a lot of time building the middle portion, which we'll call the sensing unit. And for kinase activity sensing, we pair a substrate peptide with a phosphoamino acid binding domain. And the substrate peptide needs to be very specifically phosphorylated by the kinase of interest. And the phosphoamino acid binding domain is basically a module that binds to the phosphorylated form of the substrate peptide. So the idea is when the kinase is active, we phosphorylate this substrate peptide, this red ribbon here, and this phosphorylated form will fold into the binding pocket of the binding domain, thereby synthesizing a conformational change, moving these two fluorophores close together, and then we get a change in FRET. So this allows us to look at dynamic kinase activities in living systems and we and others have used this design to build lots of cars. We started with protein kinase A, the cyclic-AMP-dependent protein kinase, and this sensor is known as ACAR. And we have built a lot of cars for the AGC family kinases, and also more recently, some of the metabolic signaling molecules, such as AMPK and also mTOR complex 1. And the one for mTOR complex 1 is called TORCAR. We have also used this type of design to build sensors for phosphatases. And again, similar designs can be used for building sensors for second messengers, these small molecules that carry the message to regulate cellular functions. So one of the ubiquitous second messengers is cyclic-AMP, and shown here is our version of the indicator for cyclic-AMP, which we call IQ. And in this particular sensor, we take one of the cyclic-AMP effector molecules known as EPAC1, exchanged protein activated by cyclic-AMP1, and use the truncated version that changes conformation upon binding to cyclic-AMP and simply sandwich that between a cyan-yellow pair. And this cyclic-AMP binding induced conformational change can be translated into a change in Fret, which allows us to look at intracellular cyclic-AMP concentrations, gradients, and nanodomains. We have also used this type of design to build sensors for lipid second messengers, such as these phosphoinositides. But in this case, it's not as straightforward for building this biosensor comparing to the cyclic-AMP sensor. For example, for sensing 3-phosphorinositide, these lipid products of PI3K, we used a plugstream homology domain from AKT, which has the right affinity and specificity for sensing these lipids. However, this pH domain doesn't change its conformation that much. As you can see, this is a ligand-free form. This is a ligand-bound form. It's very similar conformation. So we couldn't just directly use this pH domain, this lipid sensing domain, as the sensing unit. So what we did is to engineer a pseudo-ligand, which is also indicated by this red ribbon here. This is basically a stretch of acidic residues, which will pre-associate with the same binding pocket as the real ligand. And when the real ligand comes in, the lipid molecules come in, it will kick off the pseudo-ligand. And that's our engineered conformational change to convert a binding event to a change in Fret. And this general design has allowed us to build sensors for these 3-phosphorinositides and PI4,5 bisphosphate, and more recently, PI3,4 bisphosphate and PIP3 separately. We can also build color variants of these different sensors, which allow us to look at multiple species in the same living systems. And this type of pseudo-ligand design has also been used for building a sensor for RAS activity. We call this RAS activity reporter RASER. And this work was done by Ryan Weeks, a Ph.D. student in the lab. So Ryan also used the pseudo-ligand-based design by using a RAS binding domain for sensing the active RAS, which is the GTP bound form of RAS. So the same idea, when in the absence of active RAS, pseudo-ligand will pre-associate with RBD, and when the real ligand, the GTP bound form of RAS shows up, will kick off this pseudo-ligand, and that engineered conformational change can allow us to sense cellular RAS activities with a Fret change. So Ryan was able to build this first generation of a RASER, and use it to look at Golgi activity, Golgi RAS activity, and has also used this to characterize some of the RAS inhibitors. As some of you probably know, RAS has been a widely pursued targets for anti-cancer therapeutics. Targeting RAS has been the holy grail for anti-cancer therapeutics, but it was very challenging to target. But more recently, based on the pioneering work by Keyvan Shokat, some RAS inhibitors have been developed, which led to FDA-approved drugs. And these are for KRAS G12C. For example, this is one of the FDA-approved drugs. And this has motivated more of the RAS inhibitor development. So Ryan reasoned this sensor allows us to characterize these inhibitors in the cellular systems to provide additional information of how these inhibitors perform in living systems. So as you can see, just some representative data here, we're looking at one of the original G12C inhibitors, which was sent to us by Keyvan Shokat's lab, and also one of the FDA-approved drugs. In both cases, we observed it will take a long time, quite a few hours, for the inhibitors to achieve maximum inhibition. So basically, the y-axis indicates the RAS activity. The lower the ratio, the lower the RAS activity. So as you can see, probably after 3 to 5 hours, we reach the maximum inhibition. So this slow inhibition, we think, is consistent with the mechanism of these RAS inhibitors. For these G12C inhibitors, they actually target the GDP bound form of RAS, so sort of the inactive form of RAS. And the cellular RAS have to cycle through the different forms to reach this GDP bound form, then can be inhibited. So that's, we think this is consistent with the slow inhibition that we observed in these studies. And Ryan has just submitted his first author paper, I think yesterday, where he built a second generation of a RAS biosensor. And in this case, he looked at some of the newer molecular glue-based RAS inhibitors and observed very fast kinetics. And those are FRET-based sensors. I won't have time to talk about the details, but at least I wanted to show quickly show you the design. In the lab, we have also can come up with additional designs to achieve higher sensitivity and super resolution imaging compatibility for these kinase activity reporters. So the X-ray series was pioneered by Soham Mehta, a project scientist in the lab, and she recently engineered an X-ray version of the PKC biosensor. And these X-ray series cars provided ultra sensitivity for sensing kinase activity. So as you can see, for reported unit, instead of using a FRET pair, we're using a single FP. And for Flink-based biosensors, these are super resolution imaging compatible biosensors. This Flink technology was pioneered by Gary Moe, a former postdoc in the lab who has started his lab at USC. I think he's going to go through the tenure process pretty soon. Gary pioneered the Flink biosensor development in the lab, and Wei Lin, a current postdoc, is our current expert on the Flink technology. Here again, without going into too much details, I would just mention, instead of using a FRET pair, we're using another pair of a fluorescent protein, which we call Flink pair, to achieve super resolution imaging of kinase activity. But one of the challenges of Flink biosensors is they are quite photobleachable for super resolution imaging, very high intensity light is typically used. So this photo bleaching problem has haunted us quite a few years. That's why I think I will spend a bit more time introducing the newest generation of cars, which we call halo cars, which allow us to achieve higher sensitivity and super resolution imaging compatibility and alleviating the photo bleaching problem. So these halo cars were pioneered by Michelle Fry, a former postdoc in the lab. Michelle has just started her lab at ETH in Switzerland in the chemistry department. So when Michelle was in the lab, she reasoned instead of using just FP as a reporting unit, using self-labeling protein known as halotag together with a organic fluorophore could provide some additional advantages. For example, these fluorophores will be brighter and more photostable, providing better signal to noise. So in her halo cars, here I'm going to highlight a car, but Michelle actually engineered a series of halo cars. For these halo cars, we're using a circularly permutated version of a halotag and sandwich that between our kinase activity dependent switch or dependent sensing unit. We engineered it in such a way when phosphorylation occurs on the substrate and these two bind to each other, then there's a conformational change occurring within this circularly permutated halotag. And that changed the local environment of this labeled organic fluorophore, which encourage this open conformation of the fluorophore. And that leads to large increase in the fluorescence intensity. So Michelle went through a few rounds of engineering, achieved or was able to obtain an ACAR or a halo ACAR that has 13-fold increase in the fluorescence intensity. And we're also using a organic fluorophore known as GF635 provided to us by Luke Levitt's lab at Genetically Farm. And as you can see by this wavelength, this is also a far red dye, so which is really shifted to the longer wavelengths, which offered some additional advantages. So because of the high sensitivity and red shifted excitation and emission, halo ACAR was able to achieve tissue imaging with high sensitivity. So in collaboration with Jerry Olafsky lab, we were able to image pKa activity in isolated pancreatic islets at the single beta cell level. And I'm gonna switch back. So this is a simple movie showing an isolated islet where we observed glucose-induced pKa activity fluctuations. So calcium imaging in this sort of isolated islet has been done, but this is, to our knowledge, this is first time we can really look at pKa activity in this tissue setting, which opens up the possibility of looking at GLP-1R induced kinase activities in this more physiologically relevant context. Also, because of the photo stability of the fluorophore and the high sensitivity, we were able to achieve super resolution imaging using STED imaging. So this is a stimulated depletion, emission depletion methodology. And here, Michelle was able to demonstrate super resolution for using STED by using a clustering targeted halo ACAR. And she was also able to observe distinct pKa activities on two adjacent clustering-coated pits. And this suggests a highly compartmentalized pKa activity on this clutter-encoded pits. And these clutter-encoded pits has emerged to be a signaling platform as well. So we're very interested in further looking at the regulation or the mechanism underlying the highly compartmentalized pKa activity on the cluster-encoded pits. Finally, because Halo ACAR is a far-red biosensor, we were able to combine with other biosensors to achieve 5-fold co-imaging, multiplex imaging by using 5 different colors to look at 5 different activities. So we're using blue to look at calcium, cyan to look at PKC activity, green to look at PKG activity, red to look at cyclic AMP dynamics, and far red for pKa activities. And also using this multiplex imaging methodology, we were able to look at the network states downstream of quite a few different pairs of the GPCR and their ligands. For example, by looking at a beta-1 AR stimulated by adrenaline, we observed beta-1 AR is coupled to both G-alpha-S pathway and G-alpha-Q pathway, stimulating calcium and PKC activity. Also, we looked at a PDGFR and observed a similar sort of a multi-coupling, coupling both to G-alpha-S and G-alpha-Q. On the other hand, at the endogenous level, GLP-1R strongly coupled to cyclic AMP and pKa downstream of the G-alpha-S pathway, but we saw very little calcium and PKC activity at this endogenous GLP-1R level. So we reasoned with this multiplex imaging capability, Halo ACAR, together with many other sensors, really allow us to look at the dynamic network state changes downstream of GPCR signaling and allow us to characterize the network behaviors in more physiologically relevant systems. So Michelle's paper has just come out online just a couple of weeks ago. And so these are the biosensors that I would like to briefly introduce. And the remaining time, I wanted to showcase how the biosensors could be used as powerful tools for looking at the spatial temporal regulation of cell signaling. Shown here are some of our recent studies. Qi, as I mentioned, engineered an X-ray version of a PKC sensor, and that allowed him to look at how PKC is regulated at ER and lysosome. And Yonghong, a couple of years ago, using this type of approach, discovered downstream of several GPCRs, ERK is actually activated at the endosome rather than on the plasma membrane. And Danielle, Yonghong has started his lab at GIST in South Korea. And Danielle, a former postdoc who has started her lab at UCLA, used a similar approach to look at how AMPK is regulated at the lysosome and mitochondria. And more recently, Mingyuan and Aisha, 2 PhD students, designed the phosphorinositide indicator and targeted them to the lysosome and discovered these lipids are also accumulating on the lysosomal membrane. And there they are locally regulating downstream signaling molecules such as AKT and mTOR complex 1. And this work is currently in press. Today, I will focus on one study. This just recently got accepted, but it's not showing up online yet. So this work is related to cyclic AMP pKa signaling. And this is a ubiquitous second messenger kinase signaling pathway that regulate many different cellular processes. In the classical model, GPCR, if it's coupled to G alpha S, it will lead to the GPCR stimulation will lead to activation of a trimeric G protein, and the G alpha S will dissociate from beta gamma and activate adenylyl cyclase. And this is the enzyme that produces cyclin MP, which is a second messenger that sort of carried the message forward to regulate downstream signaling. And when the cyclin MP is produced inside the cells, one of the major effector molecules is pKa. So cyclin MP will bind to the regulatory subunit of the tetrameric pKa holoenzyme, unleashing the catalytic subunit. And this is the active kinase that will go on phosphorylate hundreds of different substrates to regulate many different cellular processes. Cyclin PKA are sort of the signaling molecules that link many different GPCRs to lots of different downstream processes. So one of the important questions is how this signaling pathway achieves signaling specificity. A few years ago, Zhang, a former student in the lab who is about to start his own lab, I think at UCLA, who's doing a postdoc with David Baker. When Jason was in the lab, he discovered one of the regulatory subunit of protein kinase A, R1 alpha, undergoes a liquid-liquid phase separation process to form these R1 alpha liquid droplets or R1 alpha biomolecular condensates. And these condensates sequester cyclin MP and also sequester catalytic subunit, as Julia recently showed. And that allow a highly specific signaling to be achieved for this pathway. And interestingly, an oncogenic fusion protein that formed between DNAGB1 and a catalytic subunit of pKa potently inhibit this phase separation process. And that leads to hyperactive cyclin MP-pKa pathway and leading to the tumor genesis phenotype. So this work has been published. So today I'm going to talk about a new piece of study which focuses on R1 beta. So Jason and Julia's work has been focusing on R1 alpha, and Emily, a current PhD student, has worked on R1 beta. Here's Emily. Emily shows R1 beta, just like R1 alpha, also undergoes liquid-liquid phase separation process. As you can see, this is with endogenous R1 beta with immunostaining. We can see there are puncta forming, and with phosphocholine Abimax stimulation, this is activating adenylyl cyclase and blocking PDE, basically stimulating lots of cyclin MP production. And that leads to an increase in puncta formation. And with overexpressed R1 beta labeled with a GFP molecule, we can see using recovery after photobleaching, FRET, fluorescence recovery after photobleaching, we can bleach the puncta and monitor how soon the recovery occurs. As you can see, the recovery occurs pretty rapidly, suggesting the molecules within the puncta can freely exchange with the molecule outside the puncta. And this is additional evidence for this liquid-liquid phase separation or the liquid behavior of these condensates. And here's more data with the fluorescent protein labeled R1 beta. Again, with phosphocholine Abimax stimulation, we observe more puncta formation quantified here. And with these still images, you can probably see two puncta merge together, suggesting there's no membrane separating them. Again, evidence for this biomolecular condensate formation. In vitro, using purified R1 beta that we obtained from Fritz Herberger's lab in collaboration with Susan Taylor, we show purified R1 beta can form these liquid droplets at a couple micromolar concentration. And Emily also performed truncational studies similar to R1 alpha. If we delete this N-terminal dimerization and docking domain, this DD domain, we abolish the puncta formation. Emily was able to also show if she perturbed either of the cyclin P binding domains, she lost this cyclin P induced puncta formation, suggesting the multivalency contribute to R1 beta phase separation, and the cyclin P binding domains, both of the binding domains are critical for cyclin P responsiveness. However, different from R1 alpha, R1 beta shows some diverse, or versatile condensate formation behavior. Here, Emily deleted this intrinsically disordered linker region that link the DD domain and the first cyclin P binding domain. Originally, we expect getting rid of this linker or this disordered linker, we may lose the phase separation behavior. That's what we observed in R1 alpha. However, in the R1 beta case, deleting this linker region, which Emily called delta linker, generated fibers inside the cells. As you can see, these are two cells, and pretty much all the R1 beta molecules or delta linker R1 beta molecules are in these fibers. And this is a sort of a zoom in version. We call these little sharks swimming inside the cytosol. And this is with the delta linker mutant alone. If we co-express the catalytic subunit of pKa, we can convert this fiber back to the liquid droplets. And now they can also recover pretty well, meaning the molecules can exchange between the puncta and the cytosol. This is in the presence of the cat. But in the fiber state, we saw very little recovery, suggesting they're more gel-like or even solid-like. So R1 beta is a very versatile molecule that can undergo these different sort of phase separation behavior switch. This properties also turns out to be important for this disease. So about 10 years ago, a point mutant was discovered to be causative for this neurodegenerative disorder. These patients exhibit Parkinson-like symptoms. And we can, at the cellular level, we also observe these neuronal inclusions of R1 beta, and there's also lots of neuronal loss. So this is a point mutant, and this mutation occurs in the DD domain. This triggered this following study where Emily characterized this behaviors of the disease mutant. And we are able to observe this R1 beta L50R mutant shows enhanced condensate formation. In addition, these condensate exhibit aberrant behavior. One of the main features is you can see we see very little recovery after the photobleaching. So this is the blue shows the wild type behavior, the pink shows the disease mutant. So this suggests to us these condensate formed by the L50R mutant exhibit gel-like or solid-like behaviors. This reminded us of many other examples where proteins found in pathological aggregates could undergo liquid-liquid phase separation. And the dysregulation of these condensates is linked to their respective disease states. So we asked the question, how this particular pathological assembly is linked to this physiological counterpart, and how abnormal function emerge from these aberrant condensates. So I won't go into too much details, just to show a few major pieces of data. Here, I wanted to summarize the amylase data to mention this gel-like condensate of L50R basically is able to trap catalytic subunit of a pKa potently. So two pieces of evidence are shown here, but there's more data in amylase manuscript. For example, if you look at a catalytic subunit partition into the condensates, the L50R, in the L50R case, we see significantly increased partition of the catalytic subunit. And in addition, the mobile fraction of the cat within the R1 beta puncta is significantly decreased in the L50R case. Even after the stimulation, there's a little bit there's some increase, but still lower than the wild type situation. We think the cat is trapped by the L50R R1 beta through a non-canonical pKa holoenzyme formation, which was observed by Julia in her previous study. So trapping of the catalytic subunit in these R1 beta condensates led to significant signaling defect. Using our X-ray ACAR that I mentioned previously, we observed cytosolic pKa activity was dramatically decreased. So here we're using isoproternal as a physiologically relevant stimulant to stimulate pKa activity. In the wild type situation, we saw a transient pKa activity, but in the L50R situation, we saw very little pKa activity. And we also performed RNA-seq experiments to compare the wild type and the L50R situation. And L50R cells exhibit hallmarks linked to neurodegenerative processes, such as cell stress and apoptotic signatures. Finally, in collaboration with Stefan Strack at University of Iowa, we were able to show L50R induced defects in neurite outgrowth. In fact, a lot of the neurodegenerative disorders show defect in neurodevelopmental processes. Here, again, for looking at a neurite outgrowth, we saw L50R neurons exhibit a significant defect. So to summarize this part of the presentation, we show the disease mutant L50R condensates have very different material properties, exhibit gel-like properties, and that trap the signaling enzyme, leading to a reduction of the cell signaling. And interestingly, in this case, we saw too little signaling causing neurodegenerative disease or linked to neurodegenerative disease. But in the R1 alpha oncogenic fusion protein situation, we saw too much pKa signaling that leads to tumor genesis phenotype. So very interesting yin-yang relationship here. Finally, coming back to the biochemical activity architecture, we talked a little bit, we mainly focused on the subcellular level, maybe a little bit on the super resolution level. But I wanted just to mention this type of approach could be done at the whole organism level. This is a work done in collaboration with Rick Hugener lab a few years ago. Soham and Jin Fan used X-ray ACAR to show we can observe single cell pKa activities in awake mice. The mice were running on the treadmill, and we can observe single cell neuronal activities that can be linked to animal behaviors. So this really opens up additional opportunities to look at how activities are organized at the whole organism level. I think I mentioned that majority of the people who contributed to the work, probably except for Xin Chang and Sam, two PhD students who helped revise Michelle's paper after Michelle left the lab and started her lab. In collaborators, I mentioned Fritz, Susan, and Stefan. And Pamini is our modeling expert, long-term collaborator. And Dong Wang's lab has helped a lot with the AlphaFold related studies. And finally, these are funding agencies. And I stop here and thank you for your attention. Sorry, I lost my voice.
Probably you're giving too many seminars. That's the reason. Last word.
Probably because of trends.
Yeah. Any questions, any one of you in the audience? I have one question myself. Normally, the biosensors in the biosensors area, you have an immobilized material on some sort of a surface, and then you use that actually to pass the analyte which you are trying to analyze, and then the interaction between the two of them is transduced into a signal which you measure. But here, I presume that you are actually doing most of the studies in solution. Is that correct?
Yes. So, well, solution in the sense, so we're doing all of these studies in cells, in living cells.
Mhm.
So, a lot of the sensors were expressed in cytosol. So you can you can think about it as more of a solution or a kind of viscous solution, if you will. And then in some cases, we also targeted the sensor to membranes. So plasma membrane, mitochondrial membrane, or endosomal membrane. So in that case, it may be a little bit like the situation that you described. So sort of sitting on the membrane, and then the analyte could pass by and get sensed.
Any questions for anybody else, from anybody else?
Thank you for the comment. I love it. Thanks. Optical car.
Do the companies that you're getting financial assistance from trying to, do they plan to commercialize the findings that you made in the lab so that they can make some sort of a wearable biosensor or something like that?
So we have a few patents, and some of them got licensed. But the companies that we received some support were mainly interested in using our sensors for characterizing their compounds, for developing some assays for their in-house testing, or simply wanted to support some biomolecular condensate work. So we haven't really partnered with companies to commercialize them, to the large scale yet.
That's good. It's great work, actually. Very, very, very interesting and fascinating work, actually. If there are no further questions, it's my pleasant duty to thank Zhang, Professor Zhang, for presenting a wonderful seminar. And we are sure that once we circulate this to our members throughout the country, to the ACS, there will be a lot more questions and a lot more interesting observations and all that. So I'm pretty sure that Jin will answer those questions or take a look at the suggestions and all those things. So thank you very much, Jin, for your scintillating seminar. And I thank our Scalex and our chair, Veronica Yarmello, for actually overseeing the seminar. Thanks, everybody. Bye now.
Thank you. Thanks, everyone.